paleontologists find and research ancient life forms all the time. Normally we love that. Who doesn't love a prime dinosaur fossil, right? But there's one type of ancient sample that get a lot of folks real nervous. Ancient microbes. Recently, in the Journal of Geophysical Research Biogeosciences, a research group successfully resurrected and studied 40,000-year-old bacteria from the Alaskan permafrost. What's the point of this research, and is it even a good idea? First off, these 40,000-year-old bacteria aren't nearly the oldest ones that researchers have ever resuscitated. A few years ago, for instance, researchers took deep ocean sedimentary samples that were between 4 and 100 million years old, and they successfully revivified cyanobacteria who ate and reproduced. Millions of years old! And they just perked right up like it's nothing. By comparison, this more recent 40,000-year-old sample is just that. Recent. To get these samples, these scientists got permafrost core samples from the Permafrost Research Tunnel, which extends 360 feet into the permafrost near Fairbanks, Alaska. They then took them back to the lab to grow the microbes in the samples, sequence the genes, biology things. But a lot of folks ask about the safety concerns involved in this kind of research, particularly in the context of human infection. What would happen if these ancient bacteria escaped containment? Would our immune naivete make us particularly vulnerable? Well, plenty of bacteria don't really affect or infect humans or other animals, but yes, other bacteria in thawing permafrost in some parts of the world are pretty serious. For instance, Bacillus anthracis, a soil bacteria that causes anthrax, is in the Siberian permafrost, and particularly warm summers have correlated to increased incidence of anthrax in Siberian reindeer. One outbreak in 2016 killed more than 2,600 reindeer, possibly due to the discontinuation of a vaccination program in 2007. The same research group also found Clostridium bacteria, including the kind that causes botulism. These are, in a word, bad. And to that effect, there are risks in acquiring these samples and studying them. They are extremely well-controlled risks, and there are protocols for dealing with far more dangerous samples even than those. In fact, neither Bacillus anthracis nor Clostridium botulinum is in the highest biosafety category. There are 4 levels of biosafety when studying these kinds of hazardous organisms, where level 4 includes Ebola and other hemorrhagic fevers, for instance. On the other hand, Bacillus anthracis is typically a level 2 agent, with level 3 protocols used for larger volumes. Clostridium bacteria are biosafety level 2 agents. For reference, pretty much any human cell studied in a petri dish is going to be, at minimum, a biosafety level 2 agent by default, with plenty of normal university medical research labs studying level 2+ as well. That's to say, in terms of infection risk to humans, we've got solid protocols for even more dangerous pathogens than what's been found in thawing permafrost thus far. And honestly, if we do take the samples and learn about them in the lab, then we know what kind of risks we're dealing with. I mean, imagine if we didn't know there's anthrax-causing bacteria in the Siberian permafrost. We'd be way less prepared in case something went wrong out in the world. But infection isn't the only kind of risk that can come from ancient microbes thawing in the permafrost. In fact, our researchers studying samples from the Alaskan permafrost are gauging a completely different risk from these thawing out bacteria: greenhouse gas emissions. There are huge amounts of organic carbon in permafrost, more than the CO2 in our atmosphere now. And when they really get going, these microbes can convert that matter into CO2 and methane that can get into our atmosphere. When temperatures get somewhat above freezing, between 4 and 12 degrees Celsius, aka 39 to 54 degrees Fahrenheit, microbes in the permafrost, at least as tested from this lab, start to resuscitate and reproduce. That itself is pretty cool, and these researchers found that the microbes in the permafrost had an unusual cell membrane composition that may help it survive these freezing conditions. Different strains of bacteria started to perk up and grow at different temperatures, but when they do start to wake up, they all grew incredibly slowly. Whereas E. coli divides every 20 minutes, with the sleepy microbes of permafrost, just 0.001% to 0.01% turnover in a single day for the first month after thawing. After that initial lag, though, the frosty microbes start to pick up the pace, by month 6 seeing far higher growth rates. And that data gives us a strong picture of what we're actually working with here. As the study's lead author, Tristan Caro, points out, quote, "You might have a single hot day in the Alaskan summer, but what matters much more is the lengthening of the summer season to where these warm temperatures extend into the autumn and spring. After all, a short warm season isn't long enough, based on these data, for Alaskan permafrost microbes to really get going. It's about elevated temperatures over an extended period of time, even if it's never that hot. Remember, the lower bound of thaw temperatures used in this study was just 4 degrees Celsius. These data on growth rates will allow for more accurate predictions as to how thawing permafrost will contribute to the Arctic climate over time. And maybe, as long as the permafrost refreezes seasonally, that slow reawakening of these microbes may add a buffer so that warm summers don't immediately turn into a permafrost warming feedback loop. At minimum, this is a direction of study that can help improve climate modeling predictions because the impact of permafrost is a huge point of uncertainty right now. So, for all it might seem a little risky to play with bacteria that are 40,000 years removed from anything our immune systems have ever seen, the knowledge we get from it can be useful to public health and, in this case, to climate research. In combination with future permafrost studies in other parts of the world, we can get a better idea of whether the slow reawakening observed in this study might apply other places too, which will allow us to more accurately model the relationship between changes in permafrost and greenhouse gas emissions. Imagine, 40,000 years old, and these microbes still have things to teach us.